Hi everyone, and welcome to the next webinar in our new Plantae Presents Global Plant Science Talk series. My name is Katie Rogers, and I'm your host for today's webinar. Before we get started, I'd like to go over a few things just to make sure you get the most out of attending today's webinar. If you're experiencing technical issues, please let us know about those using the chat box or by emailing me at krogers@aspb.org. If you are having trouble connecting or need to leave the webinar early, know that a recording of this webinar will be made available along with all the associated materials within a few days. If you have questions during today's webinar, please let us know using the GoToWebinar chat box. Today's talks will each be 20 minutes, followed by 10 minutes for questions. Benjamin Swessinger, who is an ARC Future Fellow and Independent Group Leader at Australian National University, is with us today to moderate your questions and read them aloud to our panelists. This webinar series is brought to you today by Plantae, the open online community for plant scientists powered by the American Society of Plant Biologists. I'd like to give a special thank you to all of our ASPB members who are attending today. Your ASPB membership dues help support and make these webinars possible. For any of you who have not yet joined ASPB, you can join today and use the discount code PRESENTS10 to receive a 10% discount on registration. ASPB members get early access to these seminars. You can learn more about ASPB and the opportunities we provide at aspb.org. This webinar is the 6th in our new virtual research talk series in response to the closure of most universities due to the COVID-19 pandemic. Please visit our webpage for more information on this series and to sign up if you'd like to be considered as a speaker. You can find us on social media at plantae.org and at ASPB. For those of you listening to this as a recording, feel free to reach out on Twitter with follow-up questions or comments using hashtag PlantaePresents. Thanks for joining us today. I'm now going to turn it over to Benjamin to introduce our speakers and moderate today's session.
Thank you very much, Katie. So I'm Benjamin. I'm a group leader at Australian National University in Canberra. I'm excited to moderate this session today with two outstanding talks, I hope, and I think we won't be disappointed. So our first speaker today will be Doris Wagner, talking about the switch to flower information, environmental tuned developmental delay. Doris Wagner is a professor at the University of Pennsylvania. She received a PhD from UC Berkeley. The research focuses on the reprogramming of cell fate and function in the context of chromatin. You can also find her on Twitter with @DorisWagner. Thank you, Doris, for joining us today, and I'm looking forward to your talk.
All right. Thank you so much for this wonderful series. I'm going to turn my webcam off to conserve here on bandwidth. And I would like to thank ASPB and Plantae for having put together this excellent series. I've listened to all of the talks so far, and it's been a very exciting thing to do now that we're all in the different new normal that we're finding ourselves in. So what I wanted to do today is talk about one aspect of the work in my lab that we have actually thought about the longest and worked on the longest, because one of the things that is afforded in this time is to kind of take a step back and put everything in perspective. And what I'd like to do is talk about three separate vignettes, as it were, that all have to do with a temporal delay. And this temporal delay is in relation to the formation of flowers, which is a very important developmental transition, as it's important for reproductive success. So, oops, now I can't advance my slides. I think Adeline had that issue. Oh, there we go. So one of the things that was kind of very instrumental in shaping a framework for how this decision works to make either a branch or a flower is this seminal paper from Enrico Cohen's lab, in which they mapped the morphospace, as it were, of inflorescence architecture. And what's apparent from this picture here right away is that the extremes at both ends of this morphospace are probably not very good for fitness. But also that when you go slightly in from these extremes, that you get very different inflorescence architectures that might be beneficial for very different environments. So here flowers are made right away. This will be a very short time to seed set and might be beneficial in environments where the growing season is very short. On the other hand, if plants first form a variety of branches and then bear flowers, you can see immediately this allows formation of more flowers and hence more seed than this scenario here, but it will take longer to get to seed set and to complete the life cycle. So one of the factors that was on this map is leafy. It's a helix to helix transcription factor that my lab has been interested in for quite some time. And this turns out to be the factor that promotes floral fate, which was in fact shown quite some time ago by Detlef Weigel when in Elliot Meyerowitz's lab and also in his own group. He showed, for example, that leafy gain of function scenarios can even shorten the flowering for the production of flowers in trees from years to months. And I'm showing here this very nice picture of the citrus tree that was done in the Martinez Zapata lab, where you can see that in these plants, flowers form pretty much right away after just a very short time compared to the wild type right here. So this, of course, is great for breeding purposes. It's not very good for production because, as I mentioned earlier, there is a limited number of fruits and flowers that form when it happens so rapidly. So what keeps leafy in check, as it were? Why is the equilibrium not on the side of leafy in this transition? So what's the temporal delay? So in Arabidopsis, leafy already comes on during this phase here when branches are formed, and then it's triggering the flower formation by upregulation of a Matzbach's transcription factor that's called apetalin-1. Apetalin-1 is important because it's only expressed in primordia that have committed to floral fate. And work in my postdoc when I was in Elliot Meyerowitz's lab, as well as by François Prassy when he was in Detlef's lab, showed that this is a direct target of leafy. Apetalin-1 is. Now, we were interested, since leafy has such a critical role in this transition, to figure out, might there be additional factors that act downstream of leafy that help with this switch to flower formation, which happens once in annual plants and is of such critical importance to reproductive success. So we took genomic approaches, and also in parallel, François Prassy took genomic approaches to identify other direct targets that might promote this switch to flower formation downstream of leafy. And when we did this, we actually came up with quite a few interesting targets. The point that it sort of led us to eventually was a different one, though, that we had set out to figure out. And that was, we had envisioned that we would find additional factors that act downstream of leafy in parallel of AP1. But as you'll see in a minute, we get led back to AP1, as it were, again and again. So we identified, for example, this factor here, LMI2. It's a MYP transcription factor. These and additional studies showed that genetically it's placed in the leafy pathway. We mapped what the role of this MYP transcription factor was and found, to our surprise, that what it does is actually help leafy upregulate apetalin-1. Now, this was unexpected. We found yet another really interesting target, in this case, not a transcription factor, so that was exciting. It was a cytochrome P450. Another group had shown this cytochrome P450 is a gibberellin catabolism enzyme, so it breaks down gibberellin hormones. And when we studied this, we made sure that indeed we were looking at the right pathway. So for example, here we can look at mutants that either lack or have lower levels of gibberellin and also are deficient in gibberellin response. And those are indeed making fewer of these branches. So lowering the gibberellin level, as you would do if leafy upregulates this enzyme, is making the transition to flowers more rapid. Conversely, if we treat with gibberellin, this is actually has the opposite effect. There's more of these branches made. Again, the surprising finding, because gibberellin actually activates the earlier transition to cessation of vegetative development, tied in with perennials, with woody perennials, where it's common practice and also has been described in the horticultural literature to treat fruit trees with paclobutrazole to enhance flower and fruit set, an effect that can be overcome when there's a second treatment of gibberellin. So yes, gibberellin does seem to inhibit flower formation. So we were able to map where this gibberellin tied into our pathway. And what we found was that lower gibberellin levels allowed accumulation of these gibberellin sensitive enzymes, the DELLA proteins, and these were needed as cofactors to SPL proteins, SPB box transcription factors, which when they're complexed with these DELLA proteins, you probably guessed it, again, tie back to apetalin-1, which is this direct target of leafy. So what we found, in effect, were two AND logic coherent feed-forward loops. This is a mouthful. What does this mean? It means that leafy is much more effective at upregulating apetalin-1 when this cofactor has accumulated, which it also activates. And it's also more effective in upregulating apetalin-1 when these factors have accumulated. So altogether, in this feed-forward loop logic that is coherent, since they're acting in the same direction, and an AND logic, because both of these together are better than one alone, we end up with upregulating apetalin-1. Now, these feed-forward loops have a couple of characteristics. They've been described really well as network motifs in the Lawn Lab. What they do very well is act as temporal delay elements. So if you have an input that leads to upregulation of factor X, which in our case would be leafy, this factor X in turn turns on LMI2 or SPL DELLA. These then act together to turn on factor Z. What happens is that leafy accumulates starting here, while this cofactor accumulates much later. So in this sense, this is a temporal delay element. So this is one way in which leafy is kept in check. And you can see here that indeed the upregulation of apetalin-1 is much delayed relative to that of leafy. What happens during this delay, of course, is branch formation, which we showed in this paper where we looked at the role of gibberellin catabolism downstream of leafy by simultaneously activating the leafy, the SPL, and repressing the gibberellin pathway. And what we get is these funny looking plants that really have lost all ability to form branches. So the first delay is due to these two coherent feed-forward loops. What about the opposing function? Is there something that acts in antagonism to leafy? And here we turn to TFL1 or terminal flower 1, which is known to promote branch formation. So as leafy promotes flower formation, TFL1 promotes branch formation. TFL1 belongs to a small family of proteins, which also includes flowering locus T or FT. In fact, these two are probably the best studied members of this family in Arabidopsis, where they act antagonistically to mediate seasonal cue-dependent regulation of important plant traits. TFL1 presumably as a co-repressor and FT likely as a co-activator in this complex here. This is not only something that we find in Arabidopsis, they're actually important for seasonal cue regulation of important traits in many, many other plant species. But we were interested in how TFL1 might execute this role. So we studied its function and asked whether it might be functioning in the nucleus, and if so, what it might be doing there, as sort of in agreement with this proposed co-repressor role. So to do that, a postdoc in the lab, Yang Zhu, as well as in parallel findings from the Schmidt lab, led us to show that TFL1 indeed has a very dramatic role in the nucleus. It had also been implicated in other cellular compartments where it locates 2000s of loci, and it does so together with this BZIP transcription factor FD. This is important because TFL1 is not capable of binding DNA on its own. So FD brings it to the DNA, as we show here when we do a TFL1 ChIP-seq in an FD mutant, where we don't see a TFL1 recruitment. And the same is shown right here. Now, what we were interested in is how this all ties back to what I've just showed you before as an antagonist, possibly of LEAFY directly, and that's indeed what we find. TFL1 FD binds to the second exon of LEAFY, where we find evolutionarily conserved BZIP motifs. It doesn't. It does not only do that, but in addition, when we look at other genes that are also bound by TFL1 FD and are de-repressed by photoinduction, so they're actually repressed by TFL1 FD, what we find is there are additional components of the pathway that I've just showed you, AP1, LMI2, and then another AP1-like protein that's called fruitful. So this identified TFL1 FD as a central repressor hub, not only preventing leafy accumulation, but also preventing, as it were, or blocking, slowing down response to leafy, the ability to leafy, for leafy to activate this critical target AP1, but also this cofactor LMI2. So now we have 2 feed-forward loops and 1 repressor. And in fact, TFL1 was on the other axis here and is the branching factor that leads to this a really stark phenotype when it's present at really, really high levels. Now, something I didn't mention in the beginning, there's not only these 2 factors here, but there's also something called a delay factor. This was called vegetativeness or VEG, which is in fact the opposite to competency. So as plants develop, there is a different competency level to respond to the accumulation of promoting factors. And this VEG factor has remained elusive as of yet. So one way that we're currently thinking about this factor is that not only do we have leafy activating apetalin-1 and this all of this transcriptional regulation going on, but we have leafy altering hormone levels. Gibberellin is just one of the levels of hormones that it alters. And when we looked genome-wide at TFL1 targets, one of the major other group of targets besides those regulating reproductive development turned out to be endogenous signaling, and a variety of hormones are there. So what we're currently hypothesizing is that the endogenous hormone state of these axillary meristems determines the competency or the vegetativeness, the ability to respond to these promoting, floral promoting signals. All right. Now, we also found in our studies that TFL1 and FT actually have opposite activities to each other. So another way to draw this pathway is like this. So they actually compete for the ability to bind to FD and to regulate downstream targets, including apetalin-1, LMI2, and leafy. So looking at the pathway in this way, it also becomes apparent that we have very strong stimulating pathways involved here that lead to flower formation. Why then this critical role of leafy? Why don't these pathways that all go into AP1, why don't they regulate AP1 by themselves? Well, it turned out the answer to that question is the third temporal delay, and that has to do with the fact that we're dealing with chromatin. So in the context of chromatin, which is shown here, which is required for the DNA to fit in an orderly way in a very small space, the nucleus, we have this first element of compaction that is required for this storage, but also has a regulatory function, and that is the nucleosome, the DNA wrapped around the histone octamer. Now, it turns out that DNA wrapped around a histone octamer is not accessible to transcription factors. The vast majority of transcription factors cannot recognize their cis motifs when they're in the DNA here as it's wrapped around the histone octamer. They can only recognize it in the linker DNA. But there's a special class of transcription factors called pioneer transcription factors, and Ken Zaret has been one of the first that studied these and also named them pioneer transcription factor, whose ability is uniquely to be able to bind their cis motifs in a context of a nucleosome. So we asked the question whether this sort of central role of LEAFY here might be due to its ability to act as a pioneer transcription factor. And to do this, we first looked at published data, MNase data that tells you where nucleosomes map in inflorescence, and we found there indeed was a nucleosome that was situated right over the leafy binding site here at the AP1 locus. So we took this piece of DNA here, 160 base pair piece of DNA, and wrapped it around the histone octamer. And when we wrapped it around the histone octamer, this migrates here in the gel. We then asked whether when we add leafy to it in increasing concentration, can it bind to its cis motif when it's in the histone octamer? And it can be retarded in this nucleosomal EMSA, but it can't do it when its binding site has been mutated. And then this LMI2 protein that I've been talking about, the MYP transcription factor, has its binding site very close to the leafy binding site, but when we add it to this nucleosomal DNA, it cannot bind to it even at high molar excess. Now, this is in vitro. What about in vivo? So we used a reprogramming system that I developed when I was a postdoc, where we can make flowers from root explants, and we use that as our system to turn on leafy production and then look at whether or not leafy can bind to sites that are were nucleosome occupied before we turn leafy on, and it can indeed do this by correlation here. So many of the leafy binding sites have nucleosomes associated with them. We figured out that this was indeed actually due to ability to bind nucleosomes by doing something called sequential CHIP, where we immunoprecipitated first for leafy, followed by histone H3, and at the AP1 locus, which is the one that we are concerned about here, we see increased binding. So we also see that leafy can displace a histone variant. We can see that it recruits chromatin remodelers, and that it can allow LMI2 to bind to this site. So here we're looking at LMI2 recruitment without or with prior leafy induction. And when we induce leafy, we don't induce leafy, LMI2 cannot bind, but it can bind when we induce leafy. So again, the surprise here came when we looked at opening off the chromatin. So this is an assay that tells you how open the chromatin is. And everything that I showed you so far happens within hours or at least within a day. But we didn't see chromatin opening at the AP1 locus within a day. We only saw it after 5 days. And this is shown here. So what does this mean? This actually follows the induction of APEDLA1 expression. So this is in that sense consistent. But what we think is going on here is that leafy, in its unique ability as a pioneer transcription factor, needs to unlock the AP1 locus to allow these cofactors that it upregulates, such as LMI2, SPL, DELLA, and other factors, to be able to bind at this locus, and this leads to further opening and eventually upregulation of APEDLA1. So altogether, this is the third delay. It's a delay in the locus opening, which is also tying everything back to leafy in the ability to be open to open this locus up for binding. So the work that I've been talking about that is recent from my lab was conducted by Ranjin. She did just defended her PhD. She worked on the pioneer transcription factor. Yang Zhu worked on the TFL1 project, and both projects were ably supported by Sammy Klasfeld, who did the computational analysis. And I also want to thank NSF for funding for this work. And with that, I'm happy to take questions.
Thank you, Doris. Very nice talk. Enjoyed this a lot. So the first question was, so you have these 3 modes of delay. Do you know from an evolutionary perspective if they, like, which one of those came first?
In terms of the slowing things down or speeding things up?
Like instead of speeding things up was a question.
Okay, so there is some idea, at least if you look into the TFL1 FT family, that that the ancestral version of that is something that I didn't talk about. It's MFT. That was there first. That's basically what the other ones were derived off, and that the slowing down might have come secondarily.
Interesting. So another question we have is there a sequence or motive in leafies that can help us to identify other pioneer transcription factors?
Yeah, so this is not based on our work, but work by Melin Fernandez Garcia from the CanSer lab. They did a survey. They study OCT4 and FOX-A's, sort of these are mammalian pioneer transcription factors. And she did a large-scale survey and found that some of the specificity resides, or the ability to do that resides in the DNA binding domain. Basically is structural, whether or not the DNA binding domain can coexist on the DNA with the histone octamer. So this has to be a very shallow DNA binding domain that doesn't generate steric hindrance with the histone octamer. And we looked at leafy, and it actually follows this pattern very nicely. It's got a very, very shallow DNA binding domain, and we have sort of in passing looked at an RF transcription factor, etin, which has a larger DNA binding domain, and for which we also have data for public data in inflorescences for CHIP and MNase, and it looked like it was not a very good nucleosome binder. Now, this is not by any means a conclusive investigation of whether or not this trend holds in plants. It's a very small sample as of yet, but one of the things could be in the DNA binding domain. Shallow contacts of the DNA on one face.
Thank you. So question pouring in now. Let's try to take a couple more. So what is the advantage of delaying flowering? Is it to have more branches?
So the later switch to making flowers allows formation of more branches, and the advantage of that is if it's toggled just right, it allows formation of more flowers and more fruit. So you actually get a much larger seed set from that, because when you think about making flowers right away, that's basically one inflorescence with just a few positions where flowers can be made. But if those elaborate into branches, then there's so many more positions. You don't want to go all the way to the very strong branching phenotype, because then that becomes detrimental again. So there's a sweet spot there. Sorry, I can't hear you.
Sorry, I forgot to unmute myself.
Oh, sorry.
Even happens nowadays. So yeah, so the question was, so you nicely illustrated how or just answered the question why delaying of flowering is important in branching. Do you know how this relates to trees? Is there anything known about how if the same pathways are implemented in trees and how they maybe lead to branching in trees and flowering in trees?
Yeah, so actually, I mean, others are much, other groups are studying this. There's very important roles for bud set, bud flush in trees for FT. Various versions of FT are present that do that. And there are people that are studying the role of TFL1 in trees as well. And it has a similar role as opposed as opposing flower formation.
Thank you. Another question. So it's a great seminar. Got the comment. Thank you. So do you know if there are any histomodifications which help recruit leafy for the opening?
Yes. So in general, the pioneer transcription factors have not been sort of repelled by histone modifications. We checked in the tissue that we were interested in, there was H3K27 trimethylation at the AP1 locus. And also in mammals, it's known that this doesn't prevent it. It could be that other types of histone modification still might prevent it. So it could be that heterochromatin, like more heterochromatic marks like H3K9 methylation might have a detrimental effect. But nothing on that has been done in plants yet.
Cool, thank you. So last question for now. So it's a question about secondary inflorescence. So the question is, what is the evidence that the secondary inflorescence developed from the shoot apical meristem and not from the lateral meristem?
I'm not sure that I would put it quite this way. So how to best address that? So initially, so there's some interesting work by Chris Kuhlmeier that showed that there are sort of reservoir of stem cells that might be passed on to axillary meristems and meristems that form on the side. So in that sense, they can be arising from that. But when the actual node, as it were, the actual axillary meristem has initiated, the trajectory that it goes on to afterwards is actually quite independent of that, what happens at the central shoot. So that's part of the model, in fact, that was published from the Cohen lab, is that in Arabidopsis, the central shoot apex is kept in an indeterminate state, prevented from making of flowers, while axillary meristems are actually already making flowers.
Thank you very much, Professor Doris Wagner. That was really an enjoyable talk, and we will pass on the remaining question for later answering. So we move on. So thank you. We move on for the second talk of the day, which is Mathew Lewsey, another Aussie. And Mathew Lewsey will be talking about analyzing the jasmonic response network from description factor activity to phenotypic outputs. Mathew Lewsey is an associate professor at the Center for Agri-Bioscience at the La Trobe University in Melbourne. And he is co-deputy director of the ARC, Australian Research Council, a research hub for medicinal agriculture. I think that was awarded recently. Congratulations. He completed his PhD in molecular biology at the University of Cambridge and conducted postdoctoral research at the Salk Institute with Joe Ecker and at the Centro Nacional de Biotecnologia. Sorry, I totally slammed this, with Roberto Solano. His research focuses on genomic systems biology and plant science, and you can also find him on Twitter at Lewsey Lab. Thank you very much, Mathew. I'm looking forward to your talk.
Thank you, Benjamin. Now, do you guys see my screen? Have I shared this correctly?
Yes, that looks good.
Excellent. All right. Yes, as Ben just said, I am going to talk about a project we conducted analyzing the jasmonic response network from transcription factor activity to phenotypic outputs. And I will just turn off my camera as well for the sake of saving everyone's bandwidth. All right, so This has been a relatively long-term project involving a lot of people, but I must point out two key people right up front. So Mark Zander and Joe Ecker, both at the Salk Institute. We have worked together on this project very closely, and either of them could also be here presenting this work today. Now, let's move this slide over. So I could not really have asked for a better person to talk next to than Doris, having just listened to her work. We're dealing with similar concepts. I guess I'm just working on a slightly shorter timescale and different stimuli that are driving transcriptional regulation. So if there is one thing that I would like everyone to take away from this talk, particularly non-experts, it's that transcriptional regulation is a lot more complex than transcription factor binds, turns gene on, turns gene off. So when a plant perceives a stimulus, let's say a hormonal stimulus or an environmental stimulus, the plant is going to drive transcriptional regulation through some very rapid acting transcription factors. We might call these master or primary regulatory transcription factors. These respond within seconds, minutes. They're going to bind genes and change expression of those genes. Now, amongst the target genes are going to be some that encode more transcription factors. We'll call them secondary transcription factors. And so these guys then go on to bind to further genes, tertiary transcription factors amongst those, and so forth. And from this, you get this cascade of transcriptional regulation occurring following the initial stimulus. And particularly recently, there's been some great work on this from Gloria Caruzzi's lab, showing how, for example, the nitrogen response has this real cascade of transcription factor activity. Now, in addition to the cascades of transcription factors, we have something that Doris just illustrated really nicely. Many transcription factors can bind at the same site simultaneously, and they can bind really close to one another. So if you looked at her slide with LFY and LMI2 on it, their recognition motifs are within about 30 nucleotides of one another. And this is not uncommon. So crystal structure studies of enhancers, for example, have shown that multiple transcription factors can bind within the same site so close that they're physically interacting with one another. So you've got enhancers and promoters that are attracting many transcription factors. It's not necessarily that one TF binds and dominates activity there. And then lastly, you have the fact that transcriptional regulation acts within larger signaling pathways. It doesn't exist in a vacuum. So you have post-translational regulatory events, changes in phosphorylation, for example, the chromatin environment, all which feeds back upon transcriptional regulation. In the study that I'm describing today, what we have attempted to do is to understand transcriptional regulation for the hormone methyl jasmonate, jasmonic acid, jasmonate. It gets referred to in a few different ways. So jasmonate is this hormone which is most well known for its effects upon defense against herbivores, certain pathogens. It modulates secondary metabolism, and it has some effects in reproductive development as well. Now, a plant perceives jasmonate through a receptor COI1. COI1 then goes on to repress some repressive factors called JAZs. So COI1 represses these JAZs. JAZs would normally repress the activity of the master regulatory TFs in this pathway, MYC transcription factors, MYC2, 3, 4, 5. MYC2 is the most well-known and the one with the strongest activity, the clearest phenotypes. So jasmonate acts to relieve repression of the MYC transcription factors. These master transcription factors then bind to other genes. They are activatory TFs. They drive expression. And amongst those, you're going to have other secondary downstream transcription factors. In our study, what we have attempted to do is set the JA genome regulatory program in its broader context. So we started from looking at transcription factor binding events and gene expression events, but we want to set these in the context of histone modifications, effects on the global phosphoproteome and proteome. And the goal here is to organize these into a model as such that allows us to better understand, possibly predict, the way in which jasmonate signaling might interact with other signaling pathways, might cross-regulate, and also attempt to use this data to get better predictions of components that affect growth and developmental outcomes. The model that we have used here is the Arabidopsis etiolated seedling. So you can see that down in the left-hand corner of my slide there. These little guys, that one's about 2 days old. So the first thing that we analyzed in our study was the suite of targets of the master regulatory transcription factors in the jasmonate response. MYC2 and MYC3, these guys that respond within minutes to a jasmonate stimulus. What I'm showing you on here is example chip-seq data for MYC2 and MYC3, where we've taken etiolated seedlings, treated them with jasmonate, and then used chip-seq to map the genome-wide targets of MYC2 and MYC3. That's 2 hours after jasmonate treatment. And these examples show classic components or classic genes known to be involved in jasmonate responses. If you can see my mouse on here, you'll see these nice, strong, replicable peaks in the promoters of these genes. So that gives you an example of what it looks like when we generate the data, but how widely do MYC2 and MYC3 bind? Well, 2 hours after jasmonate treatment, we find that MYC2 and MYC3 bind to thousands of genes. It was around about 6,000 genes. We also find that pretty much every gene bound by MYC3 is also bound by MYC2. So they're basically binding to the same genes. And if we then compare the target genes to genes differentially expressed in response to a jasmonate stimulus, we find about a third of genes that respond in expression to jasmonate are targets of MYC2 and MYC3. So these master regulatory TFs bind about a third of the jasmonate responsive genes. But what are those jasmonate responsive genes, the genes targeted by MYC2 and MYC3? Well, what we find is that MYC2 and MYC3 target much of the jasmonate signaling pathway. So on this slide, I've got a cartoon that represents most of the jasmonate signaling pathway. And any component that is colored in orange, the gene encoding that component is a target of MYC2 or MYC3 during a jasmonate response. So when you look at this, you can see that MYC2 and MYC3 are targeting genes involved in pretty much the entire signaling pathway through biosynthesis, the nuclear regulation of gene expression, and then this goes down as far as JA response-related genes and a whole suite of secondary TFs. So MYC2 and MYC3 are binding to a huge amount of the JA signaling pathway. But I told you that they target thousands of genes, and clearly this is not thousands of genes. So what else do MYC2 and MYC3 target? Well, they're also going out and binding to a lot of genes involved in other hormone signaling pathways, creating a conduit for potential cross-regulation out from a jasmonate stimulus to other hormonal pathways. So we've analyzed what the primary response transcription factors are doing, and we find that they target the cognate signaling pathway and a lot of secondary transcription factors. The next thing that we wanted to do was understand a bit more about what happens in that second wave of transcription factor activity. So what we did was to select an additional 11 transcription factors that were targets of MYC2 and MYC3 and differentially expressed in response to a jasmonate stimulus, and map the targets of those transcription factors as well, so as to analyze what they target and how much relationship there is between different transcription factors. We found that there's a great propensity for cross-regulation between the jasmonate transcription factors. So looking on the left of the slide over here, what this map is showing you is the 11 additional TFs that we determined targets for, plus MYC2 and MYC3, and which of those TFs binds to the gene encoding the other jasmonate TFs. So you can see that MYC2 and MYC3 have edges that lead out to every other transcription factor. They're binding to all of these genes. But amongst the transcription factors, pretty much every jasmonate transcription factor targets the gene encoding other jasmonate transcription factors, suggesting that they're trying to cross-regulate expression of one another. Beyond trying to cross-regulate expression of one another, they also share a huge amount of target genes, which is what I show you on the right hand of the slide. If you look in the center, you can see the strong orange blob. This represents the proportion of shared targets between the transcription factors. MYC2 and MYC3, I already said, share a huge amount of targets, and they're very strongly orange. But you can see next to MYC2 and MYC3, there's another transcription factor, STZ, which shares a lot of targets with them. And then if you look along that diagonal axis, you find other groups pop out. For example, in the bottom left, DREB2B, RA59, ERF1 share an awful lot of target genes. And what this is suggesting to us is that there may be functional groupings amongst the jasmonate response transcription factors that are attempting to regulate the same genes, potentially working either synergistically or antagonistically to regulate them. So, having analyzed events at the transcriptional level, I've said we want to organize this into the broader context of the jasmonate response. We next looked at events at the translational and post-translational level. What we did was to analyze shotgun phosphoproteomics and proteomics of plants undergoing a jasmonate response. And we looked at wild-type plants and MYC2 mutant plants. So MYC2 mutant plants, missing that master transcription factor, are going to have a somewhat hyposensitive response to jasmonate. They're not going to react to jasmonate as strongly as they should. We discovered that MYC2 and jasmonate signaling drive a substantial amount of differential phosphorylation of RNA splicing factors. And so on this graph, I'm just showing you the number of differentially abundant proteins and differentially abundant phosphopeptides between various comparisons. I will draw your attention to the tallest bars in the center. So the wild type JA versus MYC2 JA. This is the wild type plant compared to the mutant, and both are treated with jasmonate, and we're looking at how many differential proteins and phosphopeptides exist. You can see a lot of differentially abundant proteins. This makes sense. There's a strong gene expression response. You're going to get a lot of changes in protein abundance. But there are also 939 peptides, phosphopeptides, that are differentially phosphorylated. These are strongly enriched for RNA splicing factors. They're in fact the only thing particularly enriched amongst this cohort of differentially phosphorylated peptides. There are some important individual examples of where RNA splicing is important in regulating the jasmonate response. There are a small number of very well characterized examples. But what this data suggested to us was that RNA splicing might be more widespread during the jasmonate response. It might be more important in the response. To analyze that, we used time series transcriptomics. So again, we're looking at ethylated seedlings treated with jasmonate, but we're taking samples over a 24-hour period and analyzing transcriptomes. What we're using that data to do is look at genes which express more than one isoform, more than one transcript, and discover genes where the relative abundance of those transcripts changes or switches. And that looks like the example on the right-hand side of the slide, where we're looking at a single gene, two isoforms, a blue one and an orange one. At the start, blue is the dominant isoform, orange is the minor isoform, and blue remains dominant for most of the 24-hour period, but toward the end, the abundance of orange increases relatively. That becomes the dominant isoform. What these analyses showed us was that there were 137 isoform pairs from 120 genes that had isoform abundance switching occurring during the 24-hour period. So we find alternative splicing is more widespread than initially known in the jasmonate response. And this correlates with the differential phosphorylation of a number of RNA splicing factors. So up to this point, I have described to you various layers of data, but I said at the start what we wanted to do was try and integrate these data into a somewhat more predictive model that allows us to better understand or better analyze points at which jasmonate signaling might crosstalk, cross-regulate with other signaling pathways. So to achieve this, we worked with Natalie Clark and Justin Wally. So Natalie's an awesome network biologist at Iowa State, and this is her work that I'm showing you. Natalie organized the time series transcript data that I just showed you on the last slide, the phosphoproteome and the proteome data into a network that allows us to see which components are talking to one another and to understand which components might be most important regulators, so essentially important hubs. And that's what I show you on the left in orange, green, and blue. And we use the transcription factor targeting data as a validation upon the network. Now, from a 1,000-foot view, I don't expect you to take much away, much more than seeing that these things connect to one another. But what Natalie did was to use certain features of the network to predict which were the most important components. And from that, we can rank importance and pull out subnetworks where we think jasmonate might be crosstalking to other signaling pathways. I show you an example of that on the right-hand side of the screen for STZ or ZAP10. What this subnetwork predicts is that STZ acts early to repress other signaling pathways. So STZ is a known repressive transcription factor, and it seems to act early to repress pathways involved in salt and drought responses and involved in grass and steroid signaling. The network also tells us that these pathways appear to be attempting to repress the jasmonate pathway. So the prediction we get from this is that STZ is an important point for favoring the jasmonate response, for allowing it to dominate over other signaling pathways in the event of a jasmonate response. And so, just lastly, the other thing that we used these data to do was to look at whether master regulatory transcription factors and their target genes can be used to find new phenotypic components of a signaling pathway. So this idea stems from the fact that we find that MYC2 and MYC3 target an awful lot of the jasmonate signaling pathway. And we find this for other primary response transcription factors in other hormonal systems, ethylene and ABA, other authors, other labs have shown this work. So what we find is that if you take ChIP-seq targets for MYC2 and MYC3, you rank them by their strength of binding or their confidence. The targets you find at the top of the list are all of the jasmonate signaling pathway. They're the strongest targets. But spread around those are an awful lot of components that are very strong targets, genes that are very strong targets, but that are not known jasmonate response components. And so we hypothesized that they might be uncharacterized jasmonate response components. So we took approximately the top 100 unannotated targets, so to speak, and pulled out mutants for those and conducted a very simple response assay for a jasmonate phenotype. That's shown on this slide. So when you apply jasmonate to plants, it causes stunting of root growth. On the left-hand side, you see wild-type plants without jasmonate. The right-hand side, wild-type plants with jasmonate. You apply the jasmonate, they're stunted. Here's one example that we discovered, CYP78A2, which has a hypersensitive response to jasmonate. So this is simply predicted from being a strong target of MYC2. And we found another, I think, 6 components that behave the same way. Now, finding 7 phenotypic components out of 100 mutants, perhaps initially you think this is not that impressive, but bear in mind that there are probably only about 30 known phenotypic mutants in the jasmonate response pathway, which probably took about 30 years to discover. But we can dial in on new response components using master regulatory transcription factor ChIP-seq data. So with that, I will just conclude. What I've shown you here is that MYC2 controls an extensive JA genome regulatory program. Many jasmonate transcription factors form a highly connected cross-regulatory network. And we can use a JA gene regulatory network model to predict points of crosstalk with other signaling pathways. Now, this work was just published, I think March, it was published in Nature Plants, and there was a lot more in the paper than I have time to talk about today, so I'd be obliged if you're interested, if you look at it. And I should just make some acknowledgements. So again, I must highlight Mark Zander and Joe Ecker at the Salk Institute, who did a huge amount of this study. Lin Yin in my lab is a talented PhD student who's very interested in alternative splicing. We've had some brilliant collaborators, Justin Wally and Natalie Clark, Iowa State, Roberto Solano at the Centro Nacional de Biotecnología, and then Zidbar Joseph at Carnegie Mellon and a number of other people. And we have been privileged to have funding from a number of sources. Thank you.
Thank you very much, Mathew. Another exciting talk, fitting very well with Doris' talk just before. So we have a couple of questions. So the first one was about the isoform switching, the switching of these isoforms. So is there any specific features in these genes, like I mean, on the genetic level, why or how, which could maybe show what mediates isoform switching? And also, does it lead to functional change in the encoded protein?
So those are excellent questions and things that we need to look at. So we have yet to discover any clear features that determine which genes are undergoing isoform switching. I had some suspicions around intron binding of transcription factors, but that we haven't really fleshed that out yet. As for functional changes, given 120 genes, we haven't analyzed every single one individually. It fits with general models of, for example, Jas repressors are alternatively spliced during a jasmonate response, and this can create dominant negative forms, for example, but we haven't necessarily analyzed everyone in that much detail.
Cool, thank you. So there I packed two questions into one. So there were, like, what was the motivation to use 2 days etiolated seedlings? And just in line with this, another question was kind of, how do you think the transcriptional regulation and all these responses you see are different, maybe in different tissues? So do you think there's some tissue specificity to the response you're seeing?
Yeah, those are both very good questions. The motivation behind studying the 3-day old etiolated seedlings was that, generally speaking, we're working towards a much larger hormone network. So there's a lot of work that is not yet published and I haven't presented today that we're using to build these kind of models for about 6 hormones in etiolated seedlings and get a much better understanding of how crosstalk is occurring. Jasmonate is interesting in the etiolated seedling because it has some strong interactions with light signaling, but it hasn't been analyzed perhaps in as much detail as other hormones have in etiolated seedlings. In terms of the networks and signaling varying between tissues, yeah, absolutely, it definitely happens. And so one of the directions that I'm sure we and many other people are driving is looking at individual tissues, individual cells, trying to reconstruct these networks at that level. With that said, the fact that we've gone from an etiolated seedling network, made a prediction and found a phenotype in a light-grown 7-day-old seedling suggests that if you find key components, they have important roles at different stages.
Okay. Yeah, that makes a lot of sense to me. So just to add on question to this too, so do you see the responses you observe with these? You had showed some at the end some light grown plants to show some responses. Do you see the same responses in the dark and in the light?
The same responses. So jasmonate does not drive this root growth stunting in etiolated seedlings to the best of my knowledge.
Cool. Thank you. Let's take one more before we're on the clock. So it's the last one, and I don't know what this abbreviation stands for. I hope you can figure it out. So it's about STZ. So how does STZ work? It seems to repress salt and drought responses, yet favors JA response. Does this mean JA is not helpful for plants to respond to drought?
Well, that's a good question. I'm going to say I don't know, but if I was to speculate, so jasmonate and ABA do interact quite a lot, but the interactions are complex, and so I don't think they're as straightforward as just jasmonate knocks out ABA, for example. STZ is involved in salt responses. I would speculate it's perhaps generally involved in responses to certain stresses rather than being just a jasmonate transcription factor, which perhaps illustrates the complexity of these networks and why we need to get to finer and finer resolution.
All right, this is all for today. We are just on the hours. Thank you very much, Mathew, for this exciting talk. Also, thank you to Doris Wagner for the first talk. I think there was, again, 2 outstanding talks which really fit very well together. And also nice to see that some of this findings are published. You can read them in Nature Plants, or they're also provided to you from Doris on bioRxiv, so you can read up on those too, which is really exciting. For me, for today, I hand it back to Katie. Thank you both.
Yes, thank you all for attending, and thank you again to both of our speakers. Be sure to check out our website to sign up for future Plantae Presents events. We also have a Plantae webinar this Friday that will be about how to recognize the impactful papers. So be on the lookout for that as well, and be sure to sign up. Thank you all for joining.